So my name is Rachel Seaman Varner. I'm the National Soil Health Specialist with the Natural Resources Conservation Service. And I'm based in DC with the division, the Soil Health Division. So today I'll be presenting to you on soil health resource concerns and soil health indicators. This is part of our soil health continuing education series for conservation planners and farmers. So we have four main objectives today. We will be defining soil health resource concerns and the planning criteria associated with them. We'll review the use of the in-field soil health assessment tool and identify how soil health indicators reveal the presence of resource concerns that are all associated with soil function. And we will be locating and discussing the value and limitations to soil health data and the interpretations that we have currently available. So NRCS defines a resource concern as an existing or expected degradation of the soil, water, air, plant, or animal resource base to the extent that the sustainability or intended use of the resource is impaired. And this is defined in the Field Office Technical Guide. There are four soil health associated resource concerns. Those are compaction, organic matter depletion, soil organism habitat loss or degradation, and aggregate instability. And these are all sort of worded in the negative because they're identifying it as an existing or expected degradation. So that aggregate instability, for example, is worded as an instability rather than stability. So keep that in mind as we discuss the resource concerns and how they're addressed. So compaction is a management-induced soil compaction at any level throughout the soil profile that results in reduced root depth or structure and development, plant growth, biological activity, and infiltration. So the key here is that we're looking at management-induced compaction. There are inherent properties in soil that can cause a more dense layer. But these are focused specifically on management-induced compaction. That's where we identify the resource concern. So what we're looking for is that there is no clady structure that where there, under normal circumstances, there would be a different type of structure. So that's the evidence of pressure in a part of the profile. And there's no restrictive layer. We can use roots to identify compaction, where roots might be thickened or turned to the left or right as that kind of looks like a J, or layers that exceed 300 psi at field capacity when we're measuring it with a penetrometer. Organic matter depletion is another resource concern associated with soil health. And it is, again, a management-induced depletion of any or all soil organic matter pools. So that might be labile carbon, total carbon, or nitrogen. And these result in limited soil function and the processes that support plant growth, habitat or food for soil organisms, and water and nutrient cycling. So we are looking for total soil organic matter or carbon that is increasing and being monitored, and according to approved total organic matter or soil carbon tests. So there are a number of organic matter pools, and those are grouped basically by how quickly are they decomposed or turned over. There's a labile organic matter with rapid turnover. That would be living biomass, immediately available detritus or dead plant material that's on the surface. Particulate organic matter is free and partially degraded detritus or plant material, and then that free or dissolved biomolecules that have already kind of been part of the degradation process, so they're the degradation products. On the other end of the spectrum are the more stable pools of organic matter. So they are either protected chemically or physically, and they have slower turnover compared to the more labile organic matter. So that would be particulate organic matter that's protected within plant materials or microbial cell walls, the biomolecules and degradation products that might be protected chemically with chemical structures or functional groups, and then lastly, more stable char or aromatic products of fire, like the biochar might be one example of that category. So as we think about managing all of these different pools, they have different functions in the soil, but each is essential to a healthy functioning soil.
Along that rhizosphere is a whole different pool of organic matter. It's more the labile carbon and the living organisms that compile. That's part of the organic matter too. It may not show up on your soil test. That's more humus, that's more the old carbon. It takes a long time to build that on the soil test, but today we know the importance of all the fractions of soil organic matter, even the new fractions and the living fractions of organic matter and the liquid fractions of organic matter, because they are working just as hard to build the function back into your soil, build that organic matter, build the aggregate stability that allows water and air to move through the soil.
Soil organism habitat loss or degradation is another soil health resource concern. And this is the quantity, quality, diversity, or connectivity of food, cover, space, shelter, and water within the soil system. But it's inadequate. When we have this resource concern, those are inadequate to meet the requirements of soil organisms. And we look to improve habitat for soil organisms that can be determined by monitoring a number of different biological and organism-associated indicators. This resource concern is actually new. It was added in 2019. An aggregate instability is another new soil health identified resource concern, and it was also added in 2019. This is the management-induced degradation of water-stable soil aggregates that results in reduced infiltration, reduced water holding capacity in soil habitat. It can cause increased ponding, flooding, erosion, and can lead to plant stress and depressed resilience to weather extremes. So we work to improve soil aggregate stability where water stable aggregates are present at critical levels and there's no evidence of instability or poor aggregate stability, such as surface crusting and a lack of soil structure. And we'll kind of integrate this as we talk a little bit more about the indicators.
When we talk about what an aggregate is, Rick's got one crumbling up in his hand right now, but we're going to take that larger aggregate like this and we're going to start to break it up, and all of these little pieces, as we break it up, as they kind of hold their shape in this kind of angular, smaller BB size shape, those are our aggregates. And each one of these aggregates, and all of these soil particles and aggregates that are in here, these are biologically formed in these systems. So our soil microbes, in particular our bacteria and our mycorrhizal fungi and other small organisms like that, they exude biological glues that help coat those aggregates and hold them together. And so when Rick talks about building this system, he's actually building a home for the microbes so that the microbes can do their job of building aggregates. And so we really want to think about these aggregates and building aggregates in these systems.
If we do not eliminate tillage, okay, and people constantly are tilling, all they're doing is breaking these systems down, then the mycorrhizal fungi are rebuilding that system just in time to have it wiped out again by another passive tillage. If that just keeps occurring, there's no soil health being built because all they're doing is trying to rebuild their communities.
Every time, so we're getting the net loss.
That's a net loss.
So Rick talks about aggregates, and here's some great looking aggregates right here, right? And we do our aggregate stability assessment, and we take these aggregates and we put them in water. And so this is a great place to look for biological hotspots and looking for these aggregates.
The cropland assessment really is vital first step in evaluating soil health. And we have the cropland focused one. It involves a field visit, which is essential to really observe the indicators in the field. And another important piece is the interview with a producer or the land manager that really gives a lot of insight on how critical management practices are to the status of the soil health resource concerns. And it also gives insight on producers' concerns and can advise on or provide information on field and management history. The Web Soil Survey is another great resource that has a lot of information about the inherent soil properties that can really give insight on vulnerabilities and to the specific resource concerns. And then the Conservation Assessment and Ranking Tool, CART, is how that we can integrate some of this information and come to some conclusions in the planning process. So we'll talk a little bit more about that. The in-field assessment is, like I said, really the vital first step in diagnosing soil health resource concerns. And there are a number of tools that are essential to completing this process. You do complete a field visit, so visit a working operation, bring a shovel so that you can dig a hole and can actually get in to see what's happening below the surface. You can bring a soil knife to help kind of prepare the face and investigate the structure and other things. We will talk about using water and sink strainers to assess the indicators, especially aggregate instability. And infiltration rings are another piece that can be brought into the field and used to measure infiltration, which is a vital connection and part of the assessment. And you'll be taking notes, and a camera is very helpful because you can capture some of those visual observations to continue with the assessment and recall some of the details. And then a Munsell color book can help with identifying soil color. We have the Cropland Infield Soil Health Assessment Worksheet, which you can take out if you have a printed copy or reference it as a digital copy. This is designed really as a diagnostic tool to help planners determine if soil health resource concerns exist. It's not necessarily used to compare a field to another field or a means of monitoring changes in the field over time, but it is a really important and critical aspect to assessing all of the biological, physical, and chemical processes. So the way it's laid out, you have the resource concerns identified here on the left side of the screen at the top: compaction, organic matter depletion, aggregate instability, and soil organism habitat. And you can record some details about the field location and details with the working with the client. And then the center outlines all of the resource or the soil health indicators that we can evaluate and when is the best time to do so in terms of weather or irrigation events or tillage or where in the growing season. So there are some icons there associated with each indicator, and they're kind of generally organized by observations, and then when you need a particular technique really to evaluate some of them. And then on the far right of the screen here, there is a column to check yes or no if each indicator is meeting the assessment criteria. And I also brought in here the Delaware Soil Health Card. It is very handy also and connects with the National Infield Assessment very well. It overlaps with several of the indicators, and there is descriptive ratings here and potential scoring points that this can be used as another tool to help identify resource concerns. And I also wanted to mention there is an app for downloading called Land PKS. The soil health assessment, the infield assessment, is kind of integrated into this app, and the app can help complete the assessment. It can record details, and it actually has a cool feature that if you happen to forget a color book, There is part of the application includes color panels, chips to compare soil health color or soil color. So that's one of the handy options on this application. So we'll take some time to discuss each of these indicators that it can be used in the in-field assessment to identify resource concerns. Soil cover is the percent of the soil surface that's covered by plant residues, organic mulches, and or live plants. So we look at residue cover because it is associated with risks of soil erosion, how much impact a soil surface might have to withstand in terms of rain, and how much risk of erosion there is. And it also reflects kind of the amount of food source that could be supplied to soil microbes. So these associated resource concerns are aggregate instability, organism habitat, and organic matter. And assessing this in the field can include a farmer interview. They can give management history and explanation of how residues might be or may not be managed on the property. You can take a photo to make an estimate of the cover or use another state approved method. And there also here we have linked the line intercept method, which is essentially laying a transect with a measuring tape and calculating how many intersections you have with particles of crop residue. So that can be another more quantitative method of determining soil cover. So the rating criteria, soil cover will meet the criteria if it's greater than 75% after planting, and will not meet the criteria if it's less than or equal to 75%. This is just an image of a field of seed corn stubble just before planting. And on the left-hand side is just the crop residue remaining from the corn. And then on the right, you have cover crop in there as well. So you can kind of compare what very close to 100% might look like versus lower percent soil cover. And you can start calibrating your eye as you see more make up more observations in the field. You know, might begin with doing a line intercept to get a feel for what the cropping systems look like, because they all sort of are different in the way that they appear on the surface in terms of cover. So you can start with a more quantitative approach, and then as you get more accustomed to what the cropping systems in your region look like, then the estimates can be more qualitative. So another indicator we look at is residue breakdown, and this is the rate at which the crop residue on the surface is decomposing. And it really is a reflection of the biological activity of that ecosystem. So you're looking for evidence of shredding or fragmenting and a breakdown or cycling or incorporation of crop residues. And the resource concerns here are habitat loss and soil organic matter. So completing the in-field measurements, you can look for the existing residue cover and the signs of breakdown. If tillage is present in the system, then it's really very difficult to use this indicator at all. And you want to also have enough management history that you understand how many seasons have passed since that particular rotation and the residue that appears in the field. And so how many years or seasons and how many layers of crop residue are you expecting to see? The decomposition rate will be very much associated with the carbon to nitrogen ratio of the residues. So having an idea of those crop rotations and what the CN ratio of those residues would be will influence how much breakdown you should be expecting. And then you can look at the residue color and the condition of those in the field. if you're seeing a lot of small pieces that are mixed into the surface and there's minimal identification or clearly identified crop residues, then you're meeting the criteria, and you want to assess how many cropping seasons you might have. If you have more remaining from the last cropping season, then if the residue is in large pieces or left over after planting, if it can be handled without crumbling, or you're not seeing a lot of evidence of breakdown, or you're seeing residues from 2 or more cropping seasons, then it would not meet the criteria. So you can visually assess the residue breakdown looking at natural shredding and decomposition of the residues. Are you seeing those inner fibers of the stalks, for example? Are they in smaller pieces? Is there a change in color? Do you actually have visible evidence of microbial activity, like maybe you see mycelia, or you can see, where those tissues are really breaking down? Because essentially you're not wanting a corn stalk to see its 3rd birthday. So it should be completely broken down by the 3rd rotation. And the biological activity will be really reflected very well in this kind of assessment. And again, CN ratios influence it. And ultimately, we are looking at how the residues participate and are incorporated in nutrient cycling. And here's a good image of how a corn stalk might reflect a good process of residue breakdown, where you actually have evidence of fungal associations and fungus in there physically breaking down the material. So the next indicator is surface crusts, and we're looking for crusts that form after rain or an irrigation event on the soil surface, and it really reflects weak aggregate stability. So you're looking for evidence of plates that are kind of cracked, and where you have a layer of smaller particles that have consolidated at the top and formed this crust. So visually, you can observe this after rainfall or irrigation and after a period of drying. You'll note whether the crusts are throughout the entire field, or are they only in certain areas. Are they showing, when you look at the soil surface, do you see a surface layer that's more dense, that shows where these sediments have been deposited up at the top and created this crust? You might also, if the timing isn't right for that visual observation, or you're not seeing crusting in the field, you might also note if there's poor crop emergence or an uneven stand. That might be another reflection of an issue with surface crusting. So if you have evidence of surface crusts that are less than 5%, less than 5% or equal to 5% of the field, then you're meeting the criteria, and the indicator does not meet the criteria if there is greater than 5% of the field in soil crusting. And we have a few images here of soil crusts. The upper right shows that kind of broken up, crusted plates that are fitting together at the soil surface. And then that was on the left side of the screen. On the right side of the screen, you have an actual clod that shows those particles deposited at the top, where they're pretty compacted, and you can tell that there's a layer above, like, that's abnormal for the normal soil structure. And crusting does influence water movement and infiltration, so you might see evidence of that as well. So the next indicator is ponding and infiltration, and this is where areas of the field collect or hold runoff water from other parts of the field, and it can be associated with aggregate instability and compaction. And to complete the in-field measurement, the farmer interview is important. They would understand the ponding and drainage issues in the field and be able to identify areas. You can also use your own visual observation after rainfall and irrigation. You would maybe see, if you're not seeing standing water, you can also assess evidence of crop residues that may have been moved by water, that you see deposited in a certain pattern that might reflect that there was ponding that occurred. And you might also be able to observe in the field or in recent aerial photos the patterns of ponding and infiltration. And you might also see evidence of poor crop conditions. And really, if you can do the infiltration test in the field with the infiltration rings, then the soil would infiltrate 1 inch of water in 30 minutes or less. And that would meet the criteria. If it takes more than 30 minutes to infiltrate 1 inch of water, then it would not meet the criteria. Or if you still see evidence of ponding after more than 24 hours after a rain event, that would also be an indicator that this is a resource concern, or that we have an associated resource concern. Penetration resistance is another in-field assessment, and this can be done in a number of different ways, more technical to more qualitative. But this is really reflecting, again, the management-induced compaction. So a reduction in the pore space, a degradation of the soil structure. You might see platy structure when the natural structure should be something different. And it would result in decreased root development and root depth. It might cause some distorted root growth and plant growth, and would limit the soil biological habitat and activity. So there are a number of methods to assess this. It is best to actually conduct the assessment when the soil is near field capacity. And you can evaluate multiple areas and locations in the field. That will help with some comparisons. You don't want to just assess in the tractor rows, for example, or fence lines. And you want to record depths of those restrictive layers that might be detected, and the PSI ratings using the penetrometer.
You know, we're going to walk around your field here, and we're going to look for compaction today. And compaction is one of the most severe resource concerns we experience on farms. It really causes a lot of issues environmentally, and also prevents a lot of water from infiltrating and being available for your plants. So we're going to walk around. We're going to use this penetrometer here today. Albert, if you want to just hold it up.
Cool.
So this it has markers every 3 inches here. And what we're looking for today is 150 PSI or less at the 6-inch level, and from 12 to 18 inches to be under 300 PSI. And around 300 PSI is where roots start to be inhibited and cannot penetrate the soil.
Wow, it's going pretty far in.
Going in.
That it stopped. That it hasn't
Okay,
moved.
Albert, so what do you have there? So you're at over 300 PSI. So then what you want to do here is put your finger at the soil level, and Albert, go ahead and pull the penetrometer out.
Wow.
And then we can measure here. So what do we have here?
About 12 inches.
So about 12 inches.
You can also visually assess root depth and distribution, and look for evidence of platy structure.
So what we're trying to do in soil health analysis and assessment is look at the soil structure, look for this platiness, if it exists. How strong is it? Does it break apart fairly easily? We have a turnip here, and you can see what happened to that turnip root. Kind of came down instead of having a main tap root going straight down. It did take a J-turn, as we call it. So it had a little bit of difficulty at about a 2-inch layer, 2-inch depth in the soil.
So the rating criteria for this indicator are if you see granular structure and have appropriate PSI readings, or you're seeing vertical channels and good development and root depth, then you would meet the criteria. And you would not meet the criteria if there is platy structure, or if you have PSI readings that are unacceptable, or if you see evidence of root restriction, surface ponding, or abnormal root structure. So you can assess penetration or resistance with a penetrometer, which is depicted here on the left side. It actually measures the pressure it takes to insert a tip into the soil. And there's a great extension video that walks you through it in about 5 minutes that's linked here. Other ways you can detect resistant layers with a sharpshooter and a well-calibrated foot. And another method you could use is a pin flag. Recording the actual quantitative information with a penetrometer will give you that more robust assessment, but having a lot of experience in using these other methods when a penetrometer is not available, for example, is another option. Aggregate stability is another indicator, and it is related to soil porosity again, and how well the soil is held together. How well can it resist the impact of rain and reduce or resist erosion? And these are associated, this indicator is clearly associated with aggregate instability resource concern, but also very important for soil organism habitat, soil organic matter depletion, and surface compaction. So really, aggregate stability is one of the key factors in all of the soil health resource concerns. And there are 3 methods that can be used to conduct an in-field measurement of soil aggregate stability. There's the Slake test, the strainer test, and the Jornada aggregate stability test in their stability test kit. You will meet the criteria if the aggregates remain intact for greater than or equal to 80% for the Slake test, that the strainer test reflects some consistency and stability that really stands up in the strainer, and we'll talk about that in just a second on the next couple slides. And the Jornada criteria would be a rating of 5 to 6. And it would not meet the criteria if the aggregates disintegrate, if there's less than 80% remaining for the Slake test, and I'll show you some slides of that, or if the soil breaks down and slumps into a puddle, or if you see that runoff has a lot of particles in it, or lastly, if the Jornada criteria is less than 5. So these are just 2 images that really reflect how soil health management supports aggregate stability. On the left here is a example of a soil sample that has a lot of different aggregates in different sizes. It's held together very well. You can actually visually see the aggregates and the soil particles grouped together. There's healthy roots, and on the right hand side you have more of a clod that has very little visual structure. You can't really see individual aggregates. All of those particles are compressed together but not held together very well in small aggregates. And well aggregated soil infiltrates water, provides more habitat, really has a lot of benefits. So that's what you're really ideally looking for. And if you don't have well-formed aggregates, you can often have like the crust layer. You can have issues with runoff. Infiltration is less, and it causes a cascade of all kinds of issues. So the Slake test is pictured here with these columns of water that you take a soil sample or a clod, and they are supported at the top with a wire mesh. So you are completely submerging this soil sample to reflect exposure to water and how well the aggregates will hold together.
Today we're just going to walk through a Slake test, and it's something you can use for soil health assessment on a field. And it's something you want to start with a PED, and so just a clod off the surface. And Daryl's got one, I've got one. We keep a little tray of them here so that we understand where they came from. And really it's a collection of small soil aggregates glued together. And so the glues are made by the soil biology and the plant together. And if we have adequate glues, we have an integrity of the soil aggregates. So we would have soil aggregate stability. And to test this, you need some wire, a little netting. In this case, we cut the bottom out because we like to put a lot of pressure on the PED itself. And then a tall cylinder, something that holds water and is clear. And where the term Slake comes from is when we put this PED in the water is going to come into the pore spaces, and it's going to push the air out, push it out, should bubble up to the top, and it'll create a pressure. And if the pressure is greater than the glues, it's going to start to peel off or slake off as the individual soil aggregates. So
it's important, Jay, to make sure your PEDs are dry.
yep.
And so obviously these PEDs are good and dry. And so as we move forward with the experiment, again, we're going to put the PED on the screen, and we'll put them in and see what happens.
So I'm starting to see some bubbles come to the surface, Daryl.
Right, we're displacing the air, which is in the pore spaces in the soil, and so that's coming to the surface.
So now there's a pressure being created inside,
Right.
and starting to push against these soil aggregates.
And since I was the bad guy today, I get to be the bad soil, and if we look closely, we can see what's starting to happen. And
So a little history on these. The soil that I'm at right here, it's a long-term no-till field with cover crops and with livestock integration. And Daryl, your field?
my field obviously has been heavily tilled, no livestock integration, and no cover crops, and not much diversity. And so we're starting to see the results of our management.
So the cover crops make a huge difference in themselves because you, in addition to the CO2 taken in by your cash crop, afterwards you come in with a cover crop and you have a new start of CO2 that you take into the plant, and the plant ultimately putting the carbon exudates into the soil. And so then when we have the carbon in the soil, we have the food, and the biology can do its task. If we were standing outside right now and it was raining, we can get a huge amount of soil loss.
It's a little bit difficult to carry those big water columns to do the Slake test, and the slump test may be a good option. It requires less equipment. You need to be able to expose a soil sample to water, so you might need a cup or a bowl of some sort. And we use these sink strainers that are very inexpensive. You can get them at the dollar store. Take a soil sample and fill the sink strainer, and then expose it to water like you would with the Slake test. And then you can take any surface, like for example, this image here is the back of a sharpshooter, and take that strainer, flip it over after it's been exposed to water, and observe how that soil sample holds together.
One of the other assessments that you can do when you're in the field is the slump assessment. On the infield cropland soil health assessment worksheet, this is one of the tools that we can use to evaluate water stable aggregates. So when you work on this assessment, it's nice to be able to have soil from the field, and today we're going to be taking a slump from the field behind us where we have these cover crops growing. So we dug up some of the plants and plant roots, and then we're taking aggregates off of that. And so I have this tray full of soil here. And what I did is I just took any of those aggregates that were on the plant root, and I broke them up and I put them into this cup. Ideally, I like to have about BB-sized particles. That's the size that we want to deal with when we're looking at that slump assessment, if you can have BB-sized particles. And you drop them right into that container here. And once you get them all into that container, we're going to set them in water. And for our comparison...
And so you know, we've been in this really sandy soil this week, and I also wanted a comparison with a soil from a disturbed area in this sand dish to show us a contrast. So I went and I collected some soil from a more disturbed area that we'll use as a comparison.
So we're going to put them in this water here and kind of let them sit in the water for about a minute. So after it's been about a minute, we're going to pick our strainers up and allow that free water to kind of drain out from between the pores or between the aggregates, and then we're going to dump them onto our plates. So right away you can kind of see a difference in that disturbed soil versus that undisturbed soil. So my soil is the one that came from this undisturbed system, right? We have the cover crop there, we have that soil protected, we have all those living roots in there. And so those aggregates, they actually retained their shape, right? So they held together in that water and they didn't fall apart or melt. And so that's a really good thing to see, especially in the sandy soil, because building aggregates in sandy soil is probably the most difficult thing to do in these cropping systems. What do you got there, Barry?
Well, we've got something that just kind of turned to pudding. It has no structure. It would have a hard time supporting the weight of traffic. It would erode more easily. It's more easily detached from all forces, water, whether it's water, wind, or mechanical methods. And so we just have a more fragile soil situation. Once this dries, the air and water, it's less porous, so there's less oxygen moving through, so the organisms that would be biologically providing nutrients, doing that nutrient cycling, can't function at a higher level. So nutrient cycling becomes affected. That's one of our key functions that we're looking for. And then certainly everything from water infiltration to water holding capacity are very, very key soil functions that we're already getting major improvement on, where we've been in a very low disturbance situation, and we're adding lots of organic biomass to the system.
So as I slide this back and forth, what you'll notice again is those aggregates are holding together, the soil is adhering to itself. Barry, slide yours back and forth, and what kind of happens?
Well, it just turns to pudding or like peanut butter. It has no structure whatsoever. So we've just got the mineral part of the soil and some organic matter that's free floating, and you know, it muddies up the plate, stains up the plate, and the bottom line is we've just lost a lot of function, and this is just a really good assessment, really good test to see if you're on the right track or not from your management standpoint.
Soil structure can tell you a lot about the status of the soil, and it really is a good indicator that reflects really all four of the resource concerns. Soil structure is the arrangement of the particles in various aggregates, differing in shape, size, stability, and degree of adhesion to one another. All soils have a native structure. When they are managed, the structure can change, and under management conditions, you're really ideally looking for granular structure in the surface horizon, or rather the structure fits what the official series description is. So you can visually observe soil structure as you're digging that hole and you're looking at the upper horizon and comparing that to the official series description. There are a couple of images here that show very good, stable, granular structure in both of these samples at the top. So structure would not meet the criteria if you're seeing a change in the native structure, like or it's different from a granular structure. So you might see platy at the surface, or have massive structure like that one image we had where there really isn't any individual particle held together in any kind of structure. It's just all individual, or all of the particles are in one big chunk in a massive structure. You might also not meet criteria if the structure is not matching the official series description.
So what we're looking at is different structures that you'll find in soil. So granular structure is one of the, this is a good, what we would consider a good structure, but it's basically what it sounds like. It's kind of granular. In the old days, they called it crumb. In our prairie soils, we expect 99% of the time really to find a granular structure in the surface. Usually below that, what you'll get into is probably what we call blocky. Many times they call it subangular blocky structure. And that's going to be, as you move down into the profile, you're going to start to see that kind of structure. And you're seeing it right here too. Yep. Thanks, Kent. So it's the same thing you're seeing over here. There's another structure here though I want to talk about that is really common in pretty well developed soils. And that's what we call prismatic structure. See how these columns of soil come out? That's prismatic. And so that's what that's showing is this soil is very well developed. We start out with granular structure in the surface here. If I break some out, get some roots off. But you can see the granular structure in there. It's so dry it's kind of breaking into powder too, but there's granules right there. You can see them. Then it goes to prismatic structure, and they call it parting to blocky. So what they mean is when you break it apart, this prismatic structure, it comes apart and is blocky then. So in the upper part of the prismatic column, it's prismatic parting to granular, and then as you go down, it's prismatic parting to blocky.
When we were looking at the structure, we see the nice granular structure we have on the top, and you can see a great evidence of that in the structure kit, and even just right here. But really what creates that granular structure is basically the interaction between plant roots and the biology that's in the soil.
Soil color can be used as an indicator, and it's pretty well associated with soil organic matter and the resource concern. So you can assess in-field soil color using a color book and comparing to the official series description, or you can also take under managed soil, compare to an undisturbed area nearby that might be in the same series. So what you're looking for is an obvious darker layer at the surface of the soil that would reflect a buildup of more organic matter, and that it would meet the color very similar to the official series description. If you see a lighter color at the surface, then that is indicative that there may be soil organic matter depletion, or if soil mixing is observed, or if the surface is lighter than the horizon below, that can also reflect soil organic matter depletion. And color can be a little bit difficult to evaluate unless there's a native or long-term undisturbed site nearby. Generally, if the surface is lighter, like I said, that can indicate that the resource concern may exist.
So when it's dry versus when it's wet, will you get the same
Good point.
result? And does that give you an indicator of like organic matter and
A lot
soil
of
health too, just the color of it?
Definitely. You want to do a dry color and a moist color. I think sometimes you'll see a departure on the dry color and you won't pick it up on the moist color. So the only tools you need are the shovel and a few simple things that we're showing here. These color books, they're organized into hue pages. So each page is a different hue. We just happen to know that in South Dakota, most of the time we're going to start on this, what is called the 10YR page. And that's just where most of our soils land hue-wise. And then on the the bottom across here from 1 to 5 or 6, I guess. Well, actually, they skip numbers, so 1 to 8 is the chroma, and up the side, 2 to 8 is the value. All right, so what you do essentially is you just try to find the color chip that matches your soil the closest. So you take a little piece out, and you put it behind the page, and then you just move it to.
That's pretty close. Huh?
That's pretty close.
So you can compare the color, like Stan's doing, with what they identified when they first identified this soil series, like in this county. When this is dry, they would say a 10YR42 is what was typical.
So we're down here at
3.2.
3.3 probably.
Darker.
Yeah, 3.3.
And so really what we're looking at there with color, a lot of what changed the color within the same soil series, whether it's lighter or darker, is the organic matter. And so as your soil organic matter increases, you would expect the color to get darker within our hue chart there. And so most of organic matter, a good percentage of organic matter in the soil consists of carbon, and carbon in its elemental form is dark black. And so the more of it you have, the darker the soil would be. And so that's something you can kind of use as a base if you're looking at your color and you don't know if it's good, bad, or otherwise. If you can find the typical description for that soil series, and if you're the same, great. If you're lighter, maybe that's room for improvement. And if you're darker, you're probably on the right path.
The color here is not a departure from normal. It's okay, you know. So we don't see any resource concern with soil color. Now we need to do one wet too, though.
So when you do it moist, it darkens it up just by adding the moisture to it. And so I would put this one now at a 2.2. We were at a
3.3.
3.3, and moist to be a 2.2.
You can't get any darker than that, hardly.
Yep, yeah, just one more is all darker.
So now we kind of move into more of the biological assessments, and looking at plant roots and biopores is one indicator we can use. And these are important because plant roots release exudates and stimulate microbes. They provide habitat and food and build soil or aggregates, and so actually input soil organic matter. And biopores can be those channels that are created by active biological communities, and they are preserved over time from season to season. And you often find concentrations of organic matter and carbon in those areas, and they can help provide and cycle nutrients and reflect biological activity. And they also, these pores and root channels provide opportunities for aeration and infiltration and influence drainage and water storage and availability. So this indicator is really associated with all 4 of the soil health resource concerns and can be really impactful in determining the status of the field. And again, really, this is just an observation of the biopores and roots in the field. So you would be looking at the hole that was dug in the field and observing for evidence of darker areas where there are actually root channels, or cut out pores where you can actually observe channels in the soil structure. And those may be created by older plants and roots, or earthworms, or other biological organisms in the soil.
And you can see
have pores.
Yeah, some macro pores in there from either decayed roots or earthworm activity. There's just all kinds of organic matter that's just kind of
just waiting, and so
breaking down, but look at how that's breaking down.
Right. Yeah.
So biology is doing its job.
So if you have evidence of that and you have healthy roots, you're meeting the criteria if you have evidence that the roots are stressed or distorted in any way and stunted, or they don't follow previous root channels. If you're seeing no pores that are evidence from biological activity or earthworms, then this would not meet the criteria. So we have a couple of images here to help illustrate exactly the diversity of what you might find when you're assessing roots and biopores. The upper left image is a really good image where there is a channel at that upper half of the soil clod there where you can tell that there was something carved out of that soil structure. You've got roots growing in there, several different types of roots, and some evidence of some frass in there from earthworms or other organisms. So that channel has been there and is being used by a number of different types of organisms, plants and insects or earthworms or other. The center image shows very healthy roots and development there. And then the image on the right, you can see some rye roots growing down. You can see that dark color around the root channel that reflects a buildup of organic matter and other nutrients. And there's an earthworm. There's some evidence of earthworm activity also in this image. And lastly, I wanted to mention that there's the potential to see rhizosheath depending on the crop species. If there are arbuscular mycorrhizal fungi associated with the plant roots, then you may see these collections of soil particles around the roots that cling tightly to it. And that indicates a healthy rhizosphere where there's a lot of biological activity right in the vicinity in the rhizosphere around the roots. You won't likely see this with Brassica species, but other species can show that. And that can be another reflection of healthy biological activity and roots.
We want to see some biosheaths that are attached or sticking to the roots. That's the beginning of building soil aggregates. And so we pulled this rye plant out. Keep in mind that rye is going to be a grass species. It's going to produce a lot more carbon, a lot more sugars. It's going to be more mycorrhizal. And so if we zoom in really close, even in this sand, which it's very hard to build aggregates in the sand, it's very hard to get the rhizosheaths in the sand, but if you look closely, the soil is sticking to all of these roots off of that high sugar grass plant.
So assessing biodiversity is another way to observationally assess the resource concerns. You're looking for evidence and relative abundance of earthworms. You can visually see mites and springtails and millipedes, roundworms, beetles, termites, fungal hyphae, and other organisms that would provide evidence of a diversity of biological activity and a healthy soil ecosystem. The resource concerns associated are aggregate instability, soil organism habitat and loss and degradation, and soil organic matter depletion. So you're really just visually observing how many organisms do you find or evidence of those organisms. You might see worm casts or insect frass. You might see large pores or actually visually see insects or larval insects. And you can see visually fungal hyphae and other evidence. So that image at the top with kind of the white hairy stuff that could be fungal hyphae and one indicator of biological diversity. So if you have clear evidence that there are 3 or more types of organisms or evidence of organisms that you see, then you would meet the criteria. But if you're looking at, a soil sample that is a massive sample with, or you've dug the hole and you're not seeing any biological activity that's visible, if you don't see actual living organisms like earthworms or insects, or you see no evidence of fungi or no evidence of other types of macrofauna that you can visually see, then you would not meet the criteria. So this is an example of the soil earthworm middens, and one evidence of biological diversity and activity. So earthworms will pull the residue around their mounds as protection, and you can see that here. Let me. So it's these little piles are evidence that earthworms are active in the soil surface. Sorry about that. We will start looking now at the compaction, the decision trees actually for the indicators. So when you're assessing compaction as a resource concern, the primary associated indicators that you review in the field assessment are ponding and infiltration, penetration resistance, water stable aggregates, soil structure, and plant roots. So if you have surface plating that's very obvious, that shows a compaction layer in the surface, or two or more of those indicators don't meet the assessment criteria, if that is the case, then you can decide that there is a resource concern associated with compaction. And if not, then you don't have a compaction resource concern. These are all basically set up the same way, that we take the associated indicators, look at how many meet the criteria or not, and then determine whether there is the resource concern. So for soil organism habitat loss or degradation, you look at the 8 different indicators: soil cover, residue breakdown, surface crusting, aggregate stability, soil structure, plant roots, biological diversity, and biopores, and ask the question, do 2 or more indicators not meet the assessment criteria? And if the answer is yes, those 2 or more did not meet the criteria, then you have identified a soil organism habitat resource concern. And if not, then there is no resource concern, and you would have met the criteria for most of those indicators. For soil organic matter depletion, there's a little bit higher bar for identifying the depletion resource concern, where you have the same indicators, but do 3 or more indicators not meet the assessment criteria. If that is the case, then you have a soil organic matter depletion resource concern, and if not, then you do not have that resource concern. And for aggregate stability, you have the same indicators, and the decision tree is centered around whether there is prevalent evidence of aggregate instability. If you did one of the slake test or the slump test, or you see evidence in the field that would reflect clear aggregate instability, or if 2 or more indicators don't meet the assessment criteria, then if that is the case, then you can determine that there is aggregate instability as a resource concern. So Web Soil Survey is a very helpful and credible resource. Actually, just has a wealth of information, and is integrated with CART, the Conservation Assessment Ranking Tool, and is an important part of the planning process. This is the Web Soil Survey. You can search by all sorts of location information and zoom in and identify areas and get the soil mapping and soil survey information. There's also soil health information integrated into Web Soil Survey. But there is this disclaimer that there are limitations to the soil health interpretations. They do reflect a soil's inherent variability or vulnerability based on the inherent properties. So those properties that are defined by the soil parent material or texture or other inherent properties versus the dynamic properties, which are those that change with management, like aggregate stability or structure or organic matter. Those things are dynamic and changing, and the inherent parts are more static based on, you know, the parent material or mineralogy or things that aren't affected by management. But with soil health, management really is the primary driver of whether a resource concern is present. So it really requires site-specific information on the cropping systems to properly understand and assess soil health indicators and interpretations, which is why we have this in-field assessment. And the in-field assessment is an incredible tool that allows for that information. You can glean the management histories and management information that influences it, and then also just visually assess and quantitatively and qualitatively assess indicators in the field. So Web Soil Survey does provide a lot of information, but the on-site information is very important for soil health interpretations. So we have a guidance document that's really helpful in navigating through CART. It is called the Supplemental Guidance for Soil Health Resource Concerns in CART, and we have it available in Teams in our soil health teams folders and files under CART documents. And it references the manual for CART version 2.1 from February this year, and has a soil health guide tool. So we have a couple links here to the supplemental guidance, and it's really helpful, and I will show you here. So it's set up as a spreadsheet that you can enter information into. So if you recall the indicators, they're organized as they appear in the assessment. You can determine and toggle down through a drop-down menu, identifying whether you have met the criteria or not, yes or no. And then the green boxes indicate if there's a resource concern present, and I'll show you that on the next slide. And then the blue box will show which area to select in CART. So here are the results for a couple of examples and determining the 4 different soil health resource concerns. For example, at the top, you have the soil organic matter depletion, and have selected that the soil structure, plant roots, biodiversity, and biopores are meeting the resource concerns. And with 4 no's that we did not meet the criteria for soil cover, residue breakdown, water stable aggregates, and soil color, then we have determined that there is a resource concern, and it's highlighted there in green. And then the assessment level in CART would place you in that medium category where the blue box is identified. And then we have a couple of other examples here for different circumstances. So for compaction, we didn't assess penetration resistance or infiltration, but we identified that water stable aggregates did not meet the criteria, and that soil structure and plant roots did. And so this was not identified as a resource concern, and the CART assessment level would indicate that compaction is not significant. And we have a couple other examples here for aggregate instability and soil organism habitat that just kind of show how, with different in-field assessment criteria selected, how it would break down. But this is a really helpful guidance to make some decisions in CART. So CART is set up with the existing conditions. This example is for organic matter depletion, and you have, based on the web service, whether the, and that's so that rating is integrated into CART, and we'll put it into a category of being a soil that has the inherent capabilities of accumulating soil organic matter. So if it's barely capable, moderately capable, or highly capable, that's determined by the web service. And then based on the assessments, you would put it in the non, low, moderate, or high category. And then that would determine the threshold points and help with the planning process. These. So once you have the CART system approach and the CART ranking, the example we will work with here is for organic matter depletion. We had it in a medium category, and the threshold points were 51. So as you're planning, you assess the existing practices and then determine what new practices would total those threshold points. So you could use a system that incorporates conservation crop rotation for 10 points, cover crop for 15, residue and tillage management, no-till for 25, and nutrient management. So that would total 60 points. You would meet the threshold and move forward with that. You could also add other practices like soil carbon amendment, and that will go much beyond the threshold points. So we also can assess soil health using lab indicators, and the 2019 release of the technical note with recommended soil health indicators and associated lab procedures outlines the recommended indicators and the methods that are also recommended for analysis. And this was a consensus among all across USD with ARS scientists and NRCS scientists that reviewed the standard laboratory methods at the time and determined these were the easiest to complete and the least expensive, the most sensitive and robust to management that could be used for soil health indicators. And we're working on really developing regional collaboration or calibration. So all of several projects are using these soil health indicators to these lab indicators with the inherent properties of soil and the climate impacts in the regions to determine the interpretations of these indicators. So we have a scoring model that we're working on, and developing the databases and the amount enough information and data in the regions to be able to have specific calibrations and interpretations based on the variety across the country. The activity we have is called soil health testing, and it is a conservation evaluation and monitoring activity. And it supports financial assistance and technical assistance for soil health testing. And planners can use it to conduct or have a lab analyze a suite of soil health indicators. And these indicators are directly associated with specific soil functions and are a helpful addition to assessing soil health across an operation with the in-field assessment. So these two can work together. The lab indicators are quantitative. They actually have a measured value, but the interpretation is based on the relative performance of that indicator across the country and under different soil and climate conditions. And then the in-field assessment is more qualitative with some quantitative aspects to it as well. And those coupled together really provide a complete picture for soil health management. The 5 indicators that are supported by the soil health testing activity 216 are aggregate stability, soil organic carbon, active carbon, or and that's by the permanganate oxidizable carbon method, and short-term carbon mineralization, which is measured by respiration, and that reflects microbial activity, and bioavailable nitrogen recommended. The recommended method is the acid citrate extractable protein, or ACE protein method. So these 5 indicators are the basic suite of soil health indicators, and there's also the possibility of incorporating or focusing on a single indicator, and that might include any of these 5, but also could be PLFA, for example, that would measure microbial community structure, and enzymes, any type of enzyme analysis, which would measure specific microbial activities. So these indicators are associated with those functions on the left here, and can be part of the assessment for soil structural stability and how water will partition and move through the soil profile, soil organic matter cycling, carbon as a food source for microorganisms, microbial activity specifically measured by respiration, and the bioavailable nitrogen. So that is the end of the presentation.
All right, thank you, Rachel. Any questions for Rachel on soil health indicators and resource concerns?
Yeah, I just have 2 questions on the field assessment. What is a good PSI for the soil petrometer? And when you said about for the slump test exposing the soil, do you want it soaked? How long do you want it exposed to do the slump test?
Did you hear that, Rachel?
I did. So the question is, what's an appropriate PSI for penetration, the penetrometer, and how long do you soak the slump test? And it does kind of depend. The readings on the PSI on the penetrometer are, the dials usually indicate what range you need to be in. So there's like a red portion and a green portion, and it would depend on the soil type that you're in. So working with your regional specialists can also help to give some context there, but it's built in. You can kind of see it on the dial where you start getting into a range where the reflects the compaction layer. And in terms of the slump test, that does also kind of depend. It can be 30 seconds and up to a minute probably, but depending on the type of soil and how you want it to become saturated, so you can observe how long that's taking under those soil conditions, and adjust accordingly, but essentially just saturate that sample before you flip it over.
Rachel, while we have you on the line, can you give the audience a brief rundown of the 2 100 series soil management, what used to be our CAPS?
Sure, yeah. So there has been a transition from the CAP structure for planning activities, and we have a soil health management plan. Now it is called a CPA 116 and a DIA 162. And the CPA is the conservation planning activity, It is the first 7 steps of the planning process. So that's where you do the assessments and the evaluations and determining objectives. And then the 162, the design and implementation activity, as it's called now, that is step 8 of the planning process. So you would take the CPA information as the first 7 steps and evaluating the operation and the objectives, and then determining the alternatives. And then the design and implementation steps are where you really focus in on those conservation practices. How are they going to be specifically implemented? How do you meet the criteria and design and implement that plan? So the CAP really split into 2 pieces. They are focused on the principles of soil health and incorporate those 4 of minimizing disturbance, maximizing cover, maximizing biodiversity, and maximizing continuous living roots. But essentially that old CAP split into these 2 activities that are split according to the planning process.
Thank you very much, Rachel.
Thank you, everyone. Have a great day.